Superbugs capable of everything from curing diseases to mopping up pollution have come a step closer after scientists created an artificial lifeform in a lab. The new bacterial cell, nicknamed Synthia 3.0, has fewer genes than any other bacterium, making it the most basic form of life on Earth. Its creation paves the way for microbes that can be customized with genes so they churn out clean biofuels, soak up carbon dioxide from the atmosphere or pump out vaccines in industrial quantities. Dr. Craig Venter, who led the research team, said, "I think it's the start of a new era." British scientists described the work as a remarkable tour de force, and the journal Science said it was honored to publish the research. However, the technology opens a Pandora's box of ethical problems. Critics of such synthetic biology research accuse scientists of playing God. There are also fears the technique could be abused to create the artificial biological weapon. 6 years ago, Dr. Venter, a 69-year-old Vietnam War veteran and billionaire entrepreneur, made headlines around the world when he announced he had made artificial life for the first time. To do this, he read the DNA of Mycoplasma mycoides, a bug that usually infects goats. He then regraded the DNA, stitching it together from fragments of genetic code made from 4 bottles of chemicals. This DNA was then loaded into a bacterium from a different species. This bacterium read the DNA and sprang to life as an artificial bug he affectionately named Synthia 1.0. Dr. Venter, who was instrumental in the sequencing of the human genome, has now gone a step further. By a process of trial and error, he has worked out which of the 900-odd genes in Synthia 1.0 are essential for life. This led to the creation of Synthia 3.0, which boasts the 473 genes needed to grow and reproduce. If life is defined as the ability to grow and breed without help, it makes the bug the simplest living thing. In theory, Synthia 3.0, or a similar skeleton bug, could be accessorized with genes that could revolutionize healthcare and fuel production. Dr. Venter, of the J. Craig Venter Institute in California, said some of the possibilities are still out with the realms of human imagination. He also claims it may be ultimately possible to use the technique to recreate any living organism. Asked in the past why he thought he could make a better job of designing life than God, he simply said, "Well, we have computers." Dr. Victor Pinheiro, a synthetic biologist at University College London, said, "This work is a remarkable tour de force and delivers the simplest free-living organism we know." Dr. Sriram Kosuri, a biotechnologist at the University of California, Los Angeles, described the research as quite amazing. But he cautioned, the major limitation is that this is the beginning of a very long road.